Hi everyone, welcome to my website Amazing Messiah presentation. I'm Violet Evans, a biostatistics undergraduate at the University of North Carolina at Chapel Hill, and also an NSCP scholar at the National Institute of Environmental Health Sciences, or NIEHS. I'm working in the Motsinger-Reif research group under Drs. John House and Alison Motsinger-Reif. So, a little bit about my research. It's about genes and the environment. Genetics influences how the same exposure can result in different responses across individuals, whether it's to a pollutant or to a drug. So, for example, both genetic and environmental exposures play a role in the risk of skin cancer. Some genomic variants confer a greater risk for developing skin cancer. Likewise, more environmental sunlight exposure also confers increased risk of cancer. So an example of a gene by environment interaction would be where individuals with certain genetic variants have an increased skin cancer risk compared to those without, even with similar sunlight exposures. So my project involves using different approaches of identifying likely gene candidates for a gene by environment or G by E interaction. I've been mapping them to the nearest gene and performing pathway analyses to identify some of these unique and common similarities across human health outcomes from the UK Biobank. This is important because UK Biobank is a large-scale biomedical database with over 500,000 UK participants. It holds in-depth genetic and health information, and with the help of 3 different candidate identification approaches, we can find genetic variants that are associated with some of these disease risks. It helps to inform us of locations on where to look in the genome, rather than running tests against approximately all 12 million variants in European ancestry individuals. So my project will help further investigation of genetic variants that may be useful to predict the risk of some of these outcome-related complications. And I'm using RStudio for my data analysis, and fortunately don't have any wet lab work to show. So you all should care because through gene candidate identification, we are reducing the number of tests for the gene by environment to only some of the likely candidates, and therefore we address insufficient power issues and increase our ability to find significant differences. Pathway analysis revealed that neuronal signaling and cellular signaling pathways come up repeatedly across these outcomes, while gene occupancy found top hits in brain development and signaling as likely candidates. So in summary, this work suggests that some of the locations are common G by E candidates in multiple disease outcomes. Further investigation of some of these genetic variants in PEGs or other studies with outcome-associated exposures may be useful to predict the risk of complications in the context of environmental exposures. Thank you.